Good afternoon, everyone. Welcome to second session of the day 4 of FDP organized by ISF College of Pharmacy Moga. So we wholeheartedly welcome Professor Dr. VD Rangari. Welcome you, sir, on the platform of ISF College of Pharmacy. Before proceeding further, I would like to introduce today's speaker, Professor Dr. Vinod D. Rangari. Sir is working as a professor, head and dean at Institute of Pharmaceutical Sciences, Guru Ghasidas University, Bilaspur, Chhattisgarh since about 9 years. Uh, Sir was the alumni of Department of Pharmaceutical Sciences, Nagpur University, Nagpur. He has been recipient of CSIR Senior Research Fellowship at Regional Research Laboratory CSIR Jammu for his PhD under the guidance of Dr. C.K. Atal. Dr. Rangari major field of research interest are new drug discovery through herbal drug research, standardization of crude drug and phytochemistry. Since about 2 decades, he is working on the bioavailability enhancement potential of piperine. He has been successful in development of herbal drug formulation for the treatment and management of HIV, AIDS, migraine, cancer, sickle cell anemia, and has filed 8 patents on new drug formulation on HIV/AIDS, sickle cell anemia, cancer, migraine, and other diseases. Dr. Rangari has written 6 books in his professional career: Pharmacognosy and Phytochemistry Part 1 and Part 2. Published by Carrier Publication, Nasik has become bestseller in India. Another book, HIV/AIDS and Bioactive Natural Products, has been published by Studium Press Inc. USA. He has published quality research papers in peer-reviewed national and international journals. Dr. Rangari is a versatile personality with his acumen in research, art, poetry, philosophy, and Buddhism. He was invited 6 times for the United Nations Vesak Day celebration at Bangkok, Thailand. He writes poem in Hindi, Marathi, and English, and uh, philosophical, social, and life-related aspects of humanity, and has already published his poetry in Marathi, Proverbs, and in Hindi, Safar. Dr. Rangari has also published his book entitled 84,000 Dhamkhand of Buddhism. So, sir, we welcome you at the platform of ISFCP. Welcome you, sir.
Thank you, Dr. Sant. I am thankful to the organizer of this FDP on innovation in pharmacy education and applications of artificial intelligence, enhancing teaching learning practices, which is scheduled from 23 to 28th of June 25 by ISF College of Pharmacy, Moga. I thank you, my friend, Professor Gupta sir, also for inviting me for this FDP program. I would like to little correct my student also, Dr. Sant. He has said that I'm having 9 years experience in Department of Pharmacy, Guru Ghasidas University. In fact, it is now about 14 years of experience, and perhaps this is my last talk before retirement. As I would like to tell you that on 30th of June 25, just after 4 days, I am going to retire from Guru Ghasidas Central University from my academic studies.
Sir, we are grateful that we are the part of your last, perhaps last talk.
I would like to announce one more thing over here, that this presentation, Old Molecule, New Potential, Novel Tool and Revolutionary Concept in Herbal Drug Development for Rediscovering Drug Doses and Economics of the Treatment. This is the very innovative initiative which was taken by my PhD guide, the then director of the Regional Research Laboratory, which is now known as Indian Institute of IIM, Indian Institute of the Drug Development, So there Professor Dr. C.K. Atal was a director, and it was his research which I have continued on the piperine as a wonderful molecule for the uh, drug development. So let's start with this. Dear friends, all of you in pharmaceutical sciences is aware of the drug development technologies. Towards the new drug discovery, generally recent development in drug bioavailability enhancing technologies is generally considered, and such type of methods are always found, which generally reduces the drug doses cost as well as the economics of the treatment as well as side effects. So in the search of such
Sir, you are not audible. Excuse me, sir.
Is it visible to you now?
Yes, sir. Yes, sir.
Audible भी है?
Yes, sir. Yes, sir. Definitely.
I'm sorry, you are aware that Guru Ghasidas University is having a very much fluctuation in the internet facility.
Okay, sir.
ये बायोलिबिलिटी वाला चेंज नहीं हो रहा है. तो आप कंटिन्यू रखिए, ये हो जाएगा. Well, I think now it is visible to you as well as audible.
Okay, sir. Okay, sir. Okay, sir. This is visible
So
and audible.
I have just finished my first slide, and after that it was discontinued.
Yes,
So
yes, sir.
let's once again start.
Yes, sir.
This presentation as well as the talk is about towards the new drug discovery and the drug development technology, where I am taking the innovative concept of the drug bioavailability enhancing activity with the age-old molecule. We are all aware of the bioavailability in the pharmaceutical sciences. It is a measure of the rate and extent of the therapeutically active drug that reaches to our systemic circulation. And generally, bioavailability of the orally administered drugs is decreased due to its incomplete absorption as well as the breakdown in the liver by the liver enzymes. Mostly, this is one of the presentation where it is showing that when the drug goes to our gastrointestinal tract, how it is absorbed and how it passes through the first pass effect through the liver to systemic circulations. This is also another slide which is in showing the dispersion, disintegration, disaggregation, dissolution, permeation, as well as the rate limiting states associated with bioavailability of the drugs. Oral bioavailability of the drug is generally dependent upon many of the factors, but the most important factor are the stability and the solubility of the drug, passive and active transport, and the PGP efflux and cytokinin P enzymes of the livers. When we are talking about the bioavailability, let's define bioenhancer or bioavailability enhancer. This bioavailability enhancer is an agent capable of enhancing bioavailability and bioefficacy of specific drugs with which it is combined without any typical pharmacological activity of its own at the specific dose used. There are several types of approaches for the enhancement of the drug bioavailability in the pharmaceutical sciences by using the absorption enhancing criteria, by permeability enhancing dosage form like liposomes and emulsions, prodrugs approach, particle size reduction like micronization, complexation, nanoparticulate carriers, and lipid crystalline phase, P-glycoprotein inhibitors in improving the drug delivery, as well as the use of the protease inhibitors, which generally protect the drug from the degradation in the liver. There are several types of drugs in the nature which generally show a bioavailability enhancing activity. Before starting with the piperine, let's start with some of the drugs like bile salts, salicylates, non-ionic surfactants, fatty acids, ionic surfactants, chelating agents, and swellable polymers, they show some type of bioavailability enhancing activity, but not in all cases, in only certain cases. The examples of the other approaches like prodrugs, particle size reduction, P-glycoprotein inhibitors is also utilized. I would like to now take you to the old molecule new potential concept, which was initiated in Regional Research Laboratory, Jammu in 1979-80 by my guide, Professor Dr. C.K. et al. C.K. et al. Found that in our Ayurveda and Siddha system of medicine, Trikatu is one of the drug which is the mixture of equal quantity of black pepper, long pepper, and the ginger, which are known as Piper nigrum, Piper longum, and Gingiber officinalis in botanical terms. And in India, this Ayurvedic and Trikatu. It was found in Siddha system that Trikatu was being utilized along with almost all types of drugs in Ayurveda and Siddha system of medicines. So there was a question in the mind of Director Dr. C.K. et al. As well as the other scientists that why Trikatu is being utilized with all medicines, all medicinal herbal drugs, and that initiated the research on Trikatu. We are aware that the two important components of this Trikatu is black pepper, Piper nigrum, and Piper longum, which is a long pepper. And it has been found that black pepper and long pepper component which are very prominent and in very high percentage, that is piperine. And the ginger component, third component, was Gingiber officinalis, which contained mainly the oleoresins. So research was initiated on and ginger, checking that why this drug being utilized. And for the first time, before I would like to, because I want to show some other types of molecules also in nature which have the bioavailability enhancing activity. We continue with the piperine. In nature, many of the flavonoids and the other drugs, they are having the bioavailability activities. Among the flavonoids, naringin, genistein, and of flavonoids, their glycosides show a bioavailability enhancing activity, and especially they inhibit the P-glycoprotein and cytochrome P enzymes. Among the other drugs, glycyrrhizin, which is a triterpenic glycosides, niaziridine, nitrile glycosides, and the which contains mostly the essential oil components and the cumin aldehyde, they also work by the same way by inhibiting P-glycoproteins and cytochrome P enzyme. Among the other drugs, there are some other flavonoid compounds which are cyanomenin, that also shows the bioavailability enhancing activity. And lastly, we are coming to the piperine, which is antioxidant, CNS depressant, hepatoprotective, analgesic, and above all bioavailability enhancer. It shows to some extent anti-cancer, anti-tubercular, anti-HIV, cardiovascular activities. Mostly, this is one important slide where I am dedicating this presentation to my PhD guide, Professor Dr. C.K. et al., who had also been Associate Professor in Department of Pharmaceutical Sciences, Chandigarh, Punjab University, Chandigarh. He initiated this research in Araral, Jammu in 1980s, as I said, to explore the bioavailability enhancing potential of piperine. And in the latter period of time, after publications of research papers on piperine as a bioavailability enhancer, mostly they signed MOU with Cadila Laboratories, and they have developed a very good research on anti-TB drugs for proving the bioavailability enhancement of piperine along with rifampicin anti-TB drugs. So this Piper nigrum as well as Piper longum, both are the sources of piperine, and the piperine's percentage is present to the extent of 9% these drugs. But mostly Piper nigrum is utilized as a very important spice in our country as well as all over the world, and this contains a wonderful molecule in the purest possible form because there are no alkali which are present in the higher proportion. It is only piperine which is present to the extent of 99% in this drug. So this, which is known as black pepper, or in regional name we are well aware that it is known as Kali Mirch, unripe fruits which are cultivated in South India as well as in many other countries, contains 5 to 9% of piperine. Other phenolic amide alkaloids are also present to the extent of essential oil to the extent of 2.5%, and as I said that it is having some different types of other activities when utilized alone. It is very easy to isolate piperine as there is no presence of other components which are alkaloids along with it. Very few, 0.05% of the alkaloids are present which are other than piperine. So it is just the extraction with 95% absolute alcohol, and then the processing of the alcohol with process for the extraction of the alkaloids gives us 99.9% pure, well crystalline this drug material. The reason is that drug influenced by piperine are all types of categories of the drugs, right from cardiovascular, respiratory, CNS, GIT, anti-cancer, anti-HIV drugs. They are highly influenced by the piperine in the presence. Some example includes tetracycline, vaccine, rifampicin, isoniazid, and many other compounds. Even natural compound curcumin has been coenzyme A, and many other compounds are, many other drugs are influenced. I would like to tell you one of the important fact that mostly in our curries or especially in the soups, there is a tradition that we sprinkle the black pepper powder on our soups before taking it. So this is for the purpose that almost all of the important components which are present in soup, mostly proteins, amino acids, vitamins, and important other components which are there, which may be secondary metabolites, which are absorbed with the help of this piperine, which is a bioavailability enhancer. There are several types of specific bioavailability of the nutrients. The piperine can improve the bioavailability of many nutrients like vitamin C, vitamin A, vitamin B6, beta-carotene, coenzymes, and selenium. It can help combat malnutrition associated diseases, malabsorption states, and chronic ailments. And my friends, this whole bioavailability enhancing activity piperine shows only in 10 milligram in all the formulations. So if we add 10 milligram of piperine to any of the drugs, it's going to show you the best bioavailability enhancing activity in this combination. Some of my friends they say that, Dr. Sant, I'm still audible and not disconnected.
Yes, sir. You are audible, quite audible, and we are hearing, sir. Definitely, sir.
Yeah. So let's think about the mechanism of action of this bioenhancer piperine. One of the very senior professor of pharmacology as well as the scientist of Rural Jammu, when I used to discuss with him that why piperine is showing such type of wonderful activity without showing its own activity, he used to say that piperine is a molecule, it seems that it is thinking molecule. It thinks that where it should show the activity and where it should not show the activity. So he calls him a thinking molecule. And the bioavailability mechanism of the piperine is by DNA receptor binding, modulation of cell signal transduction, inhibition of the drug efflux pump, that is P-glycoprotein 2 pump, production of glucuronic acid in intestine, which allows more active forms of the drug to enter body. It may increase the absorption of the drug in the GI tract. It inhibits the enzymes responsible for the drug metabolism, especially in the liver when the drug passes through the liver after absorption from gastrointestinal tract. The mechanism of action of piperine: oral administration of piperine in rats strongly inhibits UDP glucuronyl transferase activity. And mostly it has been found that piperine inhibits the human P-glycoproteins and cytochrome P enzymes 3A4 and perhaps many other cytochrome P enzymes. Both the proteins are expressed in enterocytes and hepatocytes and contributes to the major extent for first pass elimination of many drugs. So these are the culprits for the drug metabolism, and mostly piperine generally inhibits the degradation of the drug because of the working on the P-glycoprotein and cytochrome P enzyme. Some of the metabolizing enzymes inhibited or induced by piperine includes cytochrome 1A1, cytochrome P1B1, cytochrome P1B2, cytochrome P2E1, cytochrome 3A4. All the drugs metabolized by these enzymes would therefore be influenced by bioenhancer, for showing the bioavailability enhancing activity. So in general, piperine inhibits drug metabolizing enzymes, stimulates absorption by stimulating gut amino acid transporters of the drugs. This is one of the slides which shows that the cytochrome P enzymes, which are generally induced by the drug, are inhibited by piperines, and therefore the bioavailability enhancement of the drug takes place. This is also the same slide shown in mechanism of action in a different way with P-glycoprotein and cytochrome P. It has been found that this molecule works wonderfully not only in human beings but also in all experimental animals. In mice, rats, and humans, it shows increase in the bioavailability enhancing activity with vitamins, enzymes, proteins, normal drugs, natural products, synthetic drugs. All these are generally mostly enhanced for its bioavailability. By doing so, why I have given the name to this by that how it is reducing the cost of the treatments, economics of the treatments, is because of this fact that the cost, dose, efficacy, and toxicity, all the four things are checked by piperine by using it in the form of 10 milligram. Because of the use of the drug as a bioavailability enhancer, it reduces the drug doses. We will see that how it reduces the drug doses and to what extent it reduces the drug dose. It reduces, as the drug's dose is reduced, even the cost of the drug would also be reduced. It reduces the incidence of the drug resistance. It reduces the risk of the adverse drug reactions and side effects because the dose is reduced. And this results in the great reduction in the raw material as well as the added ecological benefits because the lesser medicinal plants would be utilized because of the reduction in the dose of the drug. Now let's take the one example which is a very prominent example and successful example in the history of the drug development in India as well as the whole world, and it has also been acknowledged by World Health Organization. That is the story of rifampicin. As I said earlier, when I have introduced Dr. Atal's slide, that time I have said that Regional Research Laboratory, which is now IIIM, Jammu, it has signed MOU with Cadila Laboratory in those days, and the research was going on. Cadila is a very important pharma industry which produces rifampicin, and this sector, they are the masters as well as the pioneers. So rifampicin is a semi-synthetic antibiotics. It is red to orange platelets from acetone with a deuteron and with pK of 1.7. It is soluble in chloroform, DMSO, ethanol, methanol, and the LD50 is 885 and 1720 orally. Mostly therapeutic dose of this drug is 450 milligrams, and therapeutic category it is antibacterial and anti-tubercular drugs. This is only one slide. I would like to show you bioequivalence of the rifampicin and rifampicin piperine, which I have taken from the studies of Cadila Laboratories. Rifampicin 450 milligram is the conventional dose of rifampicin, and no doctor can dare to reduce this drug for the treatment of tuberculosis in the patients. It is not at all possible. So when we see the conventional rifampicin 450 milligram of the drug, which is shown in the green color, we see that 450 mg goes and shows the bioavailability enhancing activity. Now, this conventional dose, if we are reducing rifampicin to 200 milligram of the rifampicin with piperine 10 milligram, you can see that the bioavailability of the drug plasma concentration of rifampicin goes exactly the same or even higher than that of the conventional dose 450 milligram. So this is a wonder piperine is doing with its presence as a 20 milligram, which is not possible even by the rifampicin 450 milligram without bioavailability enhancer. This is one of the slide when the anti-TB drug 200 milligram rifampicin with 10 milligram of piperine was launched in New Delhi, and that time Dr. C.K. et al. Was also called for this launch ceremony. So Risorin is nothing but rifampicin and piperine combination. So when this drug was launched, I was having a privilege to attend this along with Dr. Atal's family, and Dr. Atal is here. He was very old during that period of time, but still he attended this, and he was honored during that period of time by Cadila Laboratories in Hotel Lalit. It was a statement of the chairman of Cadila Pharmaceutical Limited that they were expecting the company's revenue from tuberculosis segment, which is around 6 to 8 crores, may now touch over rupees 80 crores, and it exactly happened after launching the Risorin by Cadila Laboratories. This is the legacy which I have received from my guide, Dr. C.K. et al., and which I have tried to follow in these 36 years of my academic career in different institutions. And wherever I was involved in the MPharm and PhD research, I tried to give a better trial to piperine with different types of natural products. So, with the motivation from my guide, I started working on the bioavailability enhancing effect of different types of compound. First, I have taken the Taxol. In fact, when WHO, World Health Organization, accepted the research of piperine and rifampicin combination, and even when in 1980s the research papers were published about the wonderful and surprising bioavailability enhancing activity of the piperine, that time many of the pharma industries, they have worked day and night and filed the patent on the rifampicin's bioavailability enhancing active with many different types of drugs. So most of the drugs were utilized, and I was in search that which type of drug would be available for me to work. Exactly that time the paclitaxel was not worked with piperine, so I have selected this drug when I was in a very JL Chaturvedi College of Pharmacy, Nagpur. So one of the students, I wanted to work with paclitaxel. We are all aware that paclitaxel is a potent anti-cancer agent, and it is having wonderful activity. However, the drug is having a very less proportion in the plant Taxus baccata, and it cannot be produced synthetically also because of the very high cost, and the other possibilities for the production of this drug is very difficult. So I wanted to reduce the dose of the drug by using piperine bioavailability enhancer. We have worked together. I would not take much time in explaining this, but the Taxol, which is having the same type of problem, that it is having the hepatic metabolism with cytochrome P2C8 and cytochrome P3A4, these enzymes. As in case of rifampicin, in the same way, most of the anti-cancer drugs and most of the anti-HIV drugs, they are generally metabolized in the liver because of these enzymes. So we started working on this. There had been alternative approaches for increasing the bioavailability of the Taxol, which is a fat soluble drugs. Its bioavailability is very low. There had been strategies in pharmaceuticals sciences like co-solvents, emulsion, liposome, cyclodexin, nanoparticles, microspheres, chemical analogs, prodrugs, oral administration of paclitaxel with different types of enzymes. But in all these cases, it has been found that the bioavailability of the drug is not at all enhanced. This research was having added ecological benefit because of the very less proportion of this molecule in the plant itself. So that is why we have selected this drug. And wonderfully, we have found the same type of way that piperine has reduced the dose of Taxol by 10 mg addition to paclitaxel. The conventional dose and the lesser dose, we have found that the paclitaxel and piperine with 10 mg has shown wonderful activity, bioavailability enhancing activity. So the data ensures that the piperine promotes twofold increase in bioavailability of paclitaxels and as compared to the paclitaxel alone. These are some of the slides. Another drug which I have worked on is the atazanavir, which is the anti-HIV drug. This anti-HIV drug, atazanavir, I don't know where the first slide has gone. The first slide is missing over here. So atazanavir is an integrase inhibitor there are three important inhibitors in HIV: reverse transcriptase inhibitors, protease inhibitors, and integrase inhibitors, which inhibits the integrate enzyme. So mostly the cocktail of the reverse transcriptase inhibitors, protease inhibitors, and integrase inhibitors are given to the patients of HIV for the management of the disease. So as the protease inhibitors, they themselves are the cytochrome P enzymes inhibitors, but most of the drugs which are reverse transcriptase inhibitors were worked with the piperine. So I have selected atazanavir for this. So atazanavir higher dose, which is a conventional 6.5 milligram, atazanavir low dose, which we have selected, was 3.25 milligrams. And the another drugs which we have studied along with this, we have tried to study ritonavir also, which was the inhibitor of the cytochrome P enzymes, which is the reverse transcriptase protease inhibitor. Ritonavir is a protease inhibitor. We wanted to study this anti-HIV drug atazanavir with ritonavir, with piperine, as well as along with ginger oleoresin, for checking the activity of ritonavir as a bioavailability enhancer, as a piperine as a bioavailability enhancer, and ginger as a bioavailability enhancer, which is the third component of the TRICA2. So we have found the similar types of results, especially the red graph. I would like to show that atazanavir 6.5 milligram with piperine 7.5 milligram, we have shown the spike or the very high bioavailability enhancing activity of atazanavir, but the lower dose 3.25 milligram with piperine 7.5 milligram could not show the same type of activity. So in some cases the dose may be reduced, and in some cases it may not be reduced, but it shows the very good bioavailability enhancer activity. Like now here atazanavir conventional 6.5 milligram dose, if we see this orange line, it is having a very less bioavailability. But as compared to atazanavir 6.5 milligram with piperine 7.5 milligram, we show that it is having a very high bioavailability of this drug. This is one of the synthetic drugs which have worked well along with the piperine. These are some of the other graphs. The third drug I have worked upon was the fenugreek extract, and it was specially for the anti-diabetic activity. I have tried to study this drug. Fenugreek is a very good anti-diabetic agent, but this anti-diabetic activity of fenugreek is because of most of the components, but the major component which is responsible for anti-diabetic activity is 4-hydroxyisoleucine, which is the amino acid. So generally, if we soak the seeds of the fenugreek overnight in water, and this water, which is generally golden yellow color, it is having wonderful anti-diabetic activity, and it contains a good proportion of 4-hydroxyisoleucine. So this 4-hydroxyisoleucine, which we have utilized to increase the anti-diabetic activity by reducing the dose, we have got very good results with piperine. These are some of the extracts. It has shown very good activity with anti-diabetic activity with piperine and 4-hydroxyisoleucine, as well as the total extract, water extract after soaking the drug. This is for 4-hydroxyisoleucine we have studied, and it increased the bioavailability of 4-hydroxyisoleucine. Another drug which we have studied was oleonalic acid from clove, Eugenia caryophyllus. Now, oleonic acid, which is triterpenic acid, is hepatoprotective, anti-inflammatory, anti-hyperlipidemic, and anti-tumor agent. This is found in Eugenia caryophyllus in a very good amount, and it can be isolated commercially from this. It was found that in the same way as piperine works with the other drugs, it was found that piperine worked with eugenol also in the same way for the percent inhibition of the anti-diabetic activity. So it is at the lowest level, the piperine is showing the activity. I have worked with the boswellic acid also, which is triterpenic acid, and lupeol, but unfortunately the results were negative, indicating that may not be with all the different types of triterpenic acids piperine show the similar types of results. Boswellic acid and lupeol, it failed to show the bioavailability enhancing activity, especially boswellic acid, which is the anti-inflammatory and anti-arthritic drug. An Indian system of medicine, Sallaki or Salai Gugul, is having this molecule, wonderful molecule, it is having this activity, and I wanted to increase the activity because it is not showing a very prominent and significant anti-inflammatory and anti-arthritic activity in rheumatoid arthritis. So it failed to give this type of things. Many of my students worked with me for these many projects, right from 15 years around. No, right from 25 years I'm working. Like some of the prominent students who worked, like Meha Motiwala, Prachi Shukla, Garima Jain, and Swati Prakash. One of the very important aspect that I would like to tell here, that research cannot be done alone. This was a very specific type of research in which we wanted oxidative stress and free radical biology experiments. And these were lacking when we were working in Nagpur along with Meha Motiwala, especially for anti-cancer drug Taxol. But after coming to Guru Ghazidas University, I had a collaboration with University of Kolkata's Oxidative Stress and Free Radical Biology Laboratory with Dr. Debashish Bandopadhyay, and they were having an excellent model for studying the antioxidant activity in goat heart mitochondria. So for this research, Debashish Bandopadhyay played a very important role in exploring the bioavailability enhancing potential of most of these drugs after coming over here. These are some of the research papers which were published during the period of time in the very good journals. So like this, some of the patents which were also published, as I said that when there was a news and there were research papers published in the journal in 1980s, 90s, that time most of the peoples, they were just running behind piperine for filing the patents. And the first patent was filed in 1996 by Dr. Majid, who was the founder of Sabinsa Corporation. And this Sabinsa Corporation USA is having their another outlet in Bangalore and Mysore as a SAMI Laboratories. So SAMI Laboratories has filed many of the patent on piperine as a bioavailability enhancing activity, but you will find many different types of other patents also, mostly US patents that has been filed. We have also filed the patent on paclitaxel's anti-cancer activity, but after filing the patent, in fact, we found that some of the industries, they have before us, they have also filed the patent, and we could not grant this patent because in this research, the first come first serve. If you are having the research and if you are taking lot of time for exploring, in that case the other peoples, those who are faster, may get the benefit of that. So research is a very costly things, as well as it requires the facilities, it requires the manpower, as well as the state of art facilities, and it is very much necessary in our country to explore our innovative molecules for the drug development. There are many such wonderful molecules still waiting in the Ayurveda Siddha system of medicines and Indian traditional systems, which needs exploration, and the researchers of the pharmacognosy, medicinal chemistry, pharmacology, researchers of the Ayurveda and Siddha, as well as of the professional allopathic medicines, they should come together to work hand in hand for exploring the golden molecules, right? Rather, the diamond molecules which are present. One molecule which is present in the repository of the cells of the plant is as equal to the diamond mine, because when it is explored for its medicinal activity, you can see the example of salicin to aspirin, that how multi-billion-dollar industry can be built. So my dear friends, it is very much required that such type of innovative ideas should be explored, and it should be worked upon for the different types of medicinal plants in our country. There are some of the references, and I'm sure that innovative natural products are only expected to come from nature, and we should try to explore, as not even 10% of the medicinal plant world we have explored up till now. But still, over 60% of the medicinal agents which are there in the armory of the pharmaceutical sciences are coming from these herbal drugs as a natural products. So it is necessary to work on this area of herbal drugs and explore the world of the disease and its treatment. Thank you very much.
Thank you, sir. Thank you, sir. Now I would like to invite Director, sir, for his comment. Director, sir, please.
So, good afternoon, everyone. Thank you very much, Professor Dr. Rangariji. And this is the big opportunity to each and everyone, because we know that you have given one of the special title in the first slide. I think the old to the how we can to convert in the form of the novel molecules and the useful for the pharmaceutical sciences, and you have given each and every parameter related to the herbal, and this is the very useful to the whole of the community of the pharmacy, because I know that most of the peoples, they are the connecting on the herbal drug delivery system, and either it may be pharmacology, sotex, chemistry, cognosy, each and everybody doing the work like that. So I know that you have the very long experience in the field of the herbal technology, herbal drug delivery system, and how you can utilize the things in the pharmaceutical chemistry. So obviously it will be the very helpful to each and everyone. And we know that this is the time of the artificial intelligence. Then this is the how we can apply the artificial intelligence and try to develop such types of the dosage form which have the minimum toxicity and maximum efficacy, as well as the bioavailability. So Professor Rangariji, he is the very senior person in the profession of the pharmacy, well known, dedicated, and the dynamic. So I feel that we have this types of the opportunity only the rare to interact such types of the peoples. And one of our teachers, mentor, Professor Dr. M.D. Kharyaji, first time he has given the name to me, Professor Dr. Rangari, He told that Dr. Gupta, as and when you have time, you met to Dr. Rangariji, and first time I met Professor Dr. Rangariji in the Indore. So thank you very much, sir. And you are not only a good scientist, you are a really good person, down to earth. And I hope that this types of the lecture it will be the helpful to the each and every one of the scientists, and you can utilize the technology, you can utilize the concept whatever given the Professor Dr. Rangari. This lecture is the available on our YouTube channel, so you can take benefit and you can listen in the, you can say, in the loop, one by one you can listen in the loop, then you can take the more and more benefit. So thank you very much for the whole of the core committee for provide this opportunity to interact with the Professor Dr. Rangari ji, as well as number of the scientists and number of the peoples they are the connecting one this in this FDP program from throughout the country, near about 22 states. They are registered, and those who are not registered, they will also take the benefit to go through our YouTube channel and listen the lecture. So thank you very much. I also request to all of the participants, you can send your question on the YouTube channel. Our team will try to give the answer. And if we are unable to give the answer, we can connect to the speaker. And in the last day, I will connect to the speaker to justify the things, whatever they have the ask on the YouTube channel. So thank you very much, sir.
Thank you, sir. Thank you very much. I will be ever ready to answer the questions of all the participants. You can have my number from Professor Gupta sir, and you can call me anytime for the bottlenecks in the natural product chemistry, in the researches. I would be very much happy to guide you.
Thank you, sir.
Thank you, sir. Thank you, sir. It was a vast lecture on the bioavailability of the piperine. Sir, you dedicatedly work on the bioavailability aspects of the natural product whole life. I think your research and things will be referenced in future and guide us, the future researcher, in all aspects of natural products. Oh, sir, one thing more, we will be not there at your last working day. So we wish you very happy life in future. From all your students, Dr. Vivek, Dr. Balak, and Dr. Preeti is there. We wishing you happy life in future.
Thank you, Dr. Sant. I could see Dr. Asati also and Dr. Balak also sitting over there. I could not see Dr. Preeti. I wish you all the best, Dr. Sant, Dr. Asati, Dr. Balak, Dr. Preeti, and the other students of Guru Ghansdass University, which are faculty over there. I wish you all the best for a bright career. Thank you.
Thank you, sir.
Thank you, sir. Thank you.
Thank you, sir.
Thank you very much. All the best.